Okay, so in this recording, we are going to be talking about Chapter 8, Patterns of Inheritance in the OpenStax book. So, this will be covering and talking about one of my favorite scientists, who is Gregor Mendel, and his work that he completed. Here's a picture of him. His work that he completed on pea plants and his time and what he contributed as far as inheritance goes. Genes are a section of chromosomes that carry information about our traits. They are sequences of DNA that code for proteins, and all of that sounds really boring, but hopefully you will find that this chapter is fun for you to go through and to learn more about genetics. So, Gregor Mendel, just really quick backstory history. He was a monk and he studied and taught physics, botany, several natural sciences, and he specifically was in charge of some gardens, and he noticed different inheritance traits. I don't know if it was by luck or through experimentation, but he ended up working a lot with pea plants. And before this time period, a lot of people believed in a principle called continuous variation. And there's nothing wrong with this, but it doesn't apply everywhere. So, continuous variation is when there's always a blending, a further blending of traits until you're always coming to a central medium. So, there's always a mixing. Well, Gregor Mendel noticed that with pea plants specifically, that they would have something called discontinuous variation, where it was one or the other, but there wasn't always a blend. It was just this one or that one. So it was one of just a few choices, but there was never like a blending of certain things. So this is called discontinuous variation. The reason why I bring this up is and emphasize this is I think that a lot of people understand the continuous variation where they're like, oh, you just mix things together and it comes somewhere in between. But the discontinuous, just one or the other, is one that we don't see as often in humans on the outside. And so I think, or at least we don't notice it. And so people tend to think it doesn't exist, but it does. So we're going to be talking about some of that. So Mendel noticed this regarding his pea plants. So he noticed that there were purple pea plants and there were white pea plants. And if he bred those in what's called the pea generation or the parent generation, he noticed if he took the sperm, which is like pollen on a plant, and fertilized the other plant's like egg with that, that you would end up always getting a purple pea plant. And these were both like true breeding, so that means homozygous for a trait. We'll talk about that more later. But you would always get the purple as their first filial, or like their that generation. However, there's multiple ways to breed a plant. You don't always have to have two different plants. You can have the same plant. You can get one plant can self-fertilize from different flowers or even off of the same flower. And so what he found is that when this purple flower, though, self-fertilized, this purple baby plant self-fertilized, when it did that, sometimes you would end up with a purple plant, but occasionally you would end up with a white plant. So he said, hey, somewhere in this purple plant that had a one white parent plant, somewhere in here is some kind of information that is still coding for white. Even though you can't see it in any of the offspring here, you end up seeing it in Gen 2. And so that's kind of what spurred this breeding. And he learned about dominant and recessive traits this way. So a hybrid is an individual that has both two different genes for a trait. Okay, and a purebred is an organism that has only one type of gene for a trait. So I'm going to show you some of the genes that he looked at. So for pea plants, I'm going to show you round seeds, round peas are dominant over wrinkled, but wrinkled is actually a choice. You could have a wrinkled pea. And then for seed color, you can have yellow or green, and yellow is the dominant one. And then for flower color, you could have purple or white, and purple is dominant, and white is recessive. That means it's like it gets covered up by the dominant. And then you have pod-shaped for inflated, like kind of filled all the way up, or you can have constricted or kind of wrinkly. For the pod color, you can have yellow is actually dominant over green. This is actually interesting because we usually eat green peas, right? And sometimes you would see pea pods like this, but you'd kind of more expect them to be like this, and have green pea pods. But so we tend to breed the recessives more, but the yellow one is actually dominant. For flower position, you can have flowers in the middle or at the top, and middle is actually dominant. And then for height, you can have tall pea plants or short pea plants. So I keep using the word dominant and recessive. So dominants are traits that are basically the ones that express themselves even if the other trait is present. So a dominant trait is one that whether you have two of them or even if you just have one dominant, the dominant completely covers up the recessive trait. If the dominant is present, the recessive doesn't get expressed at all. It doesn't show up. So the only way a recessive trait, meaning any of these, will show up in a population is if the dominant is not there at all. I always thought of this when I was growing up as like the shy kid and like the very loud kid in class. You know, if the loud attention getting kid in class is there, then the shy student might never say anything. But suddenly, you know, you take the really louder child out of the classroom and that shyer one might start talking and showing a lot of personality and everything, and you never knew that was there. So it's only when the recessive is completely alone and it's purebred for recessive that recessive genes show up. So we are going to look at this in a few different ways. The first way is, well, I want to point out that your body cells, which are your somatic cells, remember, have two copies of each chromosome, one from your mom and one from your dad. So there's some laws to this. There's some laws in inheritance. And one has to do with the words phenotype and genotype. So your phenotype are your observable traits on the outside that everybody can see. It's what everyone can see about you. So some examples of phenotype would be, dark skin or light skin, blue eyes, brown eyes, green eyes, short, tall, male, female. Those are all phenotypes. And phenotypes can slightly be altered by the environment to some extent. For example, I got to do some science work down in Guatemala, and one of the things that we were doing there was trying to figure out gardening practices that would provide better food for the people, because there's a huge malnutrition problem in Guatemala. So this was up with the Mayan populations that they have there. So down there, Guatemalans are, I think, statistically the shortest group of people on the planet, I think. But not only are there some genetics for shortness there, but there's also severe malnutrition. So even though the people could potentially reach a certain height, a lot of them were not meeting that height because of poor nutrition. So another example of that is people that maybe go out in the sun a lot. The sun can darken some people's skin or lighten some people's hair or whatever, where there's kind of some changing of things due to the environment. So your phenotype can change a little bit with the environment, not a whole lot, but a little bit. Your genotype generally stays the same. But a phenotype is what you look like on the outside. However, that might be different than your genotype on the inside. So, an example of that could be like in humans, being short is dominant over being tall. You wouldn't think that, but short genes are actually dominant. So I happen to be a short person, and I do have some tall, like grandparents in there, but still I ended up being really short. Well, there were enough short genes in there that they masked or hid my tall genes, if I got any, I don't know, and hid those taller genes. So I might have some tall genes, but on the outside, my phenotype is just saying short. So they don't always match each other. So if you had a yellow pea plant mom and a green pea plant father, and you crossed those two pea plants, okay? Then their first set of babies, this dominant yellow, is going to mask the green color. And all of the progeny, the babies that come from that, all pea plants, are all going to be yellow in the first generation. However, then if you breed these plants to themselves or with each other, then some are going to come back out as green because um, there's some green hidden in here, and some will get flushed out in the next generation. So let's show you how this works. So if you have little y, and big Y, the big Y's representing a dominant gene and the little y's representing a recessive gene. So dominant genes are always represented by a capital, and recessive is always a lowercase letter. So both of these are also homozygous, homo meaning the same, zygotes, so they're the same genes for the traits. So this is homozygous little y, so it was the same two letters. And then this is homozygous dominant big Y. But when you combine these two, you are going to get a little y from this parent and a big Y from that parent. So now you have something called heterozygous, meaning two different. So you have a big Y and a little y, and zygote. So heterozygote, two different alleles for a trait. And all of the babies will have a big Y from one parent and a little y from the other parent. And so all of them will be big Y, little y. So now they all have this little y in there. And then when you breed some big Y, little y's together with other big Y, little y's, you're going to get some with big Y, some with big Y, little y, and some with two little y's. So, heterozygotes like this, mixed individuals, are also called carriers, meaning they carry the trait of something else even though they're not expressing it. So this can result in different breeding things that you see come out, but also it's important for different disorders and things that exist. Sometimes people have a gene that's hidden that maybe is undesirable, maybe even something that's bad that can be a disorder, for example. So, and they don't have the disorder themselves, but they're a carrier for that disorder. That's why some people end up getting genetics tested before they have kids with somebody else, because they want to know if we're both carrying a certain disorder, what are the chances our child would get that disorder. So you can have that. So down here, I want to show you, I'm going to go, I wish they had the other picture. So we'll just look at this. Okay, so this individual with a big Y is going to be yellow. Okay, big Y is going to be yellow. But big Y is also yellow. So you can get yellow two different ways. You can have two big Y's, or you can have a big Y and a little y, and you're still going to be yellow. The only way you can be green, though, is to have two little Y's. So you can always tell the genetic makeup from the appearance. So you can't always tell the genetic makeup from the phenotype. With the recessives, you do know what they are genetically because they can only be little y's. But for the dominant traits, you don't know if they have two of the dominant alleles or a dominant and a recessive allele. So I want to talk about, it kind of makes sense here then to talk about what's called a test cross. So I wish they had a picture from that. They did not include it. So we will stay here. So with a test cross, basically geneticists, or well, it's not always done by geneticists. Sometimes it's done by conservation people. Sometimes it's done by breeders or in agriculture. So sometimes people don't know if the individual, what the genes are for a dominant individual. So they'll breed that dominant individual with a recessive allele. If there ends up being, I think I have a picture of this actually. I'm going to go here. Okay, so I'm going to show you this picture. If you have, with a test cross, if you don't know the genetics of this black sheep, so you don't know if it's big B, or big B, little b, but you do have a white sheep, and the white sheep you know is little b, because there's no other way you could have a white sheep. So you breed the white sheep with the black sheep, and you see what kind of babies come out. So if the parent, let's assume this is mom and this is dad. They both have a great grumpy face there. So say if mom is big B, and you know dad is little b, all the babies you ever get from this pairing will always be black because the mom will always give a big B, and that will always mask dad's little b white genes. So you're going to always have sheep that are black because they'll always be big B, little b. Now, if you take the same two different parents, I'm sorry, and you breed the white sheep, which is little b, dad, with this sheep, the mom, which is big B, little b, and you end up getting some white sheep out of it, you know that this mom had some little b in her, because the only way you could get that is if mom had big B, she's going to pass down her big B's and 50% will be black, but from her gene that is white, she will pass that down. Dad will pass down over his little b genes, and you'll get about 50% of the sheep babies will end up being white, and because they have a little b from mom and a little b from dad, they'll be little b, little b. So that's how they run a test cross. Test crosses are basically breeding an unknown individual with a known individual and seeing what the offspring are. And that's done in agriculture a lot. That's done out in conservation efforts, because if you have very few individuals left of a certain population, for instance, California condors have a lethal gene, a lethal dwarfism gene, where the baby will die if it gets that gene. They want to know ahead of time for the breeding, so they can look at things like test crosses to determine that. So we are going to move to the law of segregation. So I don't have a good picture of this, so I'll just talk about it. So Mendel's work led to the scientific law of segregation, which is during gamete formation, and it applies to all sexually reproducing organisms. Uh, the law states that the paired genes segregate equally and have an equal chance of getting expressed and picked as the genes. So, in other words, all genes that are available can get used equally in the offspring. So, no specific genes have a better chance of getting passed down to the offspring. So, a better way to put this is like if you were drawing names out of a hat and whoever's name got drawn out, they were going to win a prize. If you got to be the person drawing the names out of the hat, and so you crumpled your own piece of paper so that you could feel your crumpled name in the hat and pick that one, that would be an unfair advantage. Well, it doesn't work that way because of the law of segregation with genes. Each allele has equal chance of getting passed down to the offspring. Okay, so this was another picture of test cross in your book. I'm going to say with this that this, I didn't mention, is called a Punnett Square. And a Punnett Square is a grid that explains inheritance, and there's a lot of great videos online that explain this if you don't know how to do this. And I put several in our course that you can check out. Okay, so there's different laws. There's a law called the laws of inheritance, and there's some parts that go along with that we are going to talk about. Humans have about 46 chromosomes, or 23 pairs, and lots of things contribute to what are expressed as the individual's phenotype. This can include their gametes, and gametes are an individual's genes, and can even be like your gender. Most genes are on the autosomes, which are body cells, but some genes are actually linked to your sex cells. For example, the ability to see color. Some people are colorblind, and that affects males because it's a sex-linked gene more than females. And I'll let you look up a little more information about how that works. So individuals inherit one allele for each trait from each parent. However, sometimes you inherit two different traits, but one doesn't completely dominate over the other. This is called incomplete dominance. So the example here is of these snapdragons. This is an example of incomplete dominance. Now with snapdragons, red is dominant over the white color. So notice though that these flowers aren't red or white. They're kind of a blended pink, but they're a lot closer to red, right, than they are to white. So that's called incomplete dominance. It's where the dominant one gets a little washed out, but just a little bit. That's called incomplete dominance. So that white gene in there washed the red out just a little bit and made it sort of pink. Okay, there's another phenomenon called codominance. And in codominance, that's when both alleles are expressed at the same time. So in humans, this actually happens with our blood type. So you can have type A blood, which has A antigens on its outside, or you can have type B blood that has B antigens on the outside. Or if you inherited A from mom and B from dad, you can have AB blood, which has both A and B antigens on the outside. That's an example of codominance when they were both dominant. And then the last choice is you can have actually type O blood, where you don't have any antigens on the outside of your blood. So in this case, let me see if I have a better picture. if you inherit from mom, we'll say mom's genes are along the top and dad's genes are along the side. If you inherit type A blood from mom and A blood from dad, you're going to have type AA, which will give you type A blood. That makes sense, right? Well, if you get B from mom and A from dad, you're going to have type AB blood. But if you inherit type O blood, which doesn't have any factors on the outside from mom, and A from dad, the dominant A is going to override the O and you're going to have type A blood. You have type A blood. It's just that recessive one is not going to make a difference. So let's go down to here. If you have A from mom and B from dad, you're going to have type AB blood. And we talked about that right there. If you get B from mom and B from dad, you'll have type B blood. But if you get type O from mom and B from dad, you're going to have type B blood. And then I'm going to skip to this one because we've already talked about these two up here. If you get O from mom and O from dad, you're going to have type O blood. So I want to show you here that type O blood is actually pretty rare. It's pretty hard to get type O blood. You had to have the O gene from mom and the O gene from dad. I also want to say that the only blood that you know is homozygous is type O blood, because A can be heterozygous. It can be like this, and B can be heterozygous because it can be B and O. So type O blood is the only one that for sure is homozygous when you do have that kind of blood. So type or blood type is also considered a trait that's determined by multiple alleles. Like so far we've been talking about yellow or green, tall or short. Well, here's something that has multiple alleles, and yet you still have individuals that only get 2. So while there were 3 choices, A, B, and O, you still only get 2 total alleles. So that's just important to remember. In a population, the most commonly seen organism is called the wild type. So it's like what you see out in the wild is where that name came from. So most of the time, the wild type is usually the dominant trait, but sometimes you can have the wild type be a recessive trait. And how does that happen? We'll say you have a huge field of all white snapdragons, and there's just like one red snapdragon plant, and it's off to the side, so it doesn't breed much with the other plants. So the wild type is the white snapdragons, even though they are the recessive one. So sometimes the wild type is recessive, although most of the time the wild type is dominant, just because it tends to dominate. Okay, so look up sex-linked traits. This is Drosophila flies, and it's going to be important for you to learn about sex-linked traits. I'll go over it really briefly. Let me see if they have a picture. Okay, so some traits, if they are tied to the sex chromosomes, and there's not very many like this, but if they are tied to the sex chromosomes, you have to remember that males only get 1. The Y won't carry any of these chromosomes. So whatever they get on the X is what they'll have. So you can see in this test cross here, there's the red eye and the white eyes. Red eyes are the most common, and white eyes are kind of the less common eye color. But when we bred this male and female here, and the female gave her X chromosomes, and the male gave his Y chromosomes, I'm sorry, that was opposite. When the male gave his X X chromosomes and the female gave her X and the male had his Y contribution, you ended up getting some white-eyed males. So males, when something is tied to the X chromosome and they only have one X chromosome that they get, they end up having a higher chance of getting that trait. So that happens a lot with color vision in humans. So look up more on that if you are interested. Okay. So we've talked about the law of independent assortment, that genes independently assort and they don't influence each other. But I do want to point out in this section that there are some genes that are always so close to each other, they're like linked to each other. So they're very close to each other on the chromosome that when assortment happens, they just end up assorting together. So this kind of violates the law of independent assortment, which is just an important piece to remember. And then I want to give you, before we end, there's a few terms that I want to go over. I want to mention the term pleiotropic. Pleiotropic genes are defective genes that affect different phenotypes. For example, in cystic fibrosis leads to, so one gene from cystic fibrosis can make an individual have respiratory problems, can make them have salty sweat, can cause them to have vitamin deficiencies. So one gene, which, is from cystic fibrosis, is causing these very different problems in different areas. So that's one gene that has multiple influences, is called a pleiotropic gene. Then there's another term that I want to mention, which is polygenic. Polygenic inheritance is where many genes influence just one trait. For example, in humans, think about skin color. Skin color isn't just one gene from each parent. It's actually many genes from each parent. Or hair color, for example. Some research shows that hair color as high as 124 genes are influencing your hair color. So that's how you can have, a big family and multiple individuals have very different hair color, very different skin color, and they were from the same parents. You can get that because of polygenic inheritance. I also want to go over a few, let me see if I can make this a little bit bigger. Where's the presentation mode? I want to go over a few, I'll make that smaller again. I'm going to go over a few different inheritancing that I want you to see. So these are called Palomino horses, and Palomino horses are a mixture of what's called a cremello horse, which is this white color, and this chestnut color. And you get this horse that's kind of this tan color called Palomino. Well, how do you get that? So I'm going to show you how. A Palomino horse is a mixture of a big B from one parent, brown, which is dominant over little b for white. And when you bring that big B down to each square in the Punnett Square and bring the little b's over, you end up getting babies that are big B, little b all throughout, which is this Palomino color. But say you have a brown, a homozygous brown parent, so big B, and then you had a Palomino, so a big B, little b, like tan, other parent. So in that case, you end up getting some of the babies, some of the offspring have, will tend to be the chestnut brown color, and some will be Palomino. So what if you just said, well, I just want only Palominos. I love that color. I just want all Palominos. So I'm just going to just breed Palomino horses. Well, if you do that, you'll see that genetically, sometimes you get the chestnut, and sometimes genetically, you get the cremello or the white color. And only half the time do you end up getting Palomino. So the way that you get Palomino horses is to always mix the chestnut color with the cremello color to get the Palomino mix blend. Okay. You might have some questions about calculating gene frequency and how frequent a gene shows up in a population. This is actually pretty straightforward. If there's 100 individuals in a population of horses and 20 were big B, 40 were big B, little b, and 40 were little b, the way that you would figure out the frequency of little b is to just add this up. There were 40 little b, which means 20 of each. 20 of this type of b, 20 of that kind of b, which equals 40. And then there were 40 big B, so 20 of the big B plus 20 of the little b equals 40. So there was 20, 40, 60 little b's. And then of the big B's, it said that there were 20 big B's here, so 10 here and 10 here equals 20. And then there were 20 big B's here, so 20 plus 20 is 40. So there were 60 little B's and 40 big B's. Okay, so that would be the gene allele frequency. I want to look at some human traits really fast. One is cleft chin. I want you to look at some of these. A cleft chin is actually dominant in humans. If you have a smooth chin or if you have a cleft chin, cleft is actually dominant over smooth, so you can look at your own and see what you have. I don't know why people are always nervous about doing these. A widow's peak is where you have pointy hairline, and sometimes it's just a little pointy, but sometimes it is way smooth, and that's called like a natural hairline or a smooth hairline. So anyways, look and see what you have. I think I have a slight widow's peak. It is not as smooth as this. And then the next one is having an attached or a free or detached earlobe. So some people have their earlobe where it's almost like stuck to the side of their head. And some of us are like flying free with like a very free flappy earlobe, which this is me. I'm the free flappy earlobe. And sorry, I have a dog that is sneezing and moving in the background. I apologize for that. But that detached earlobe is the dominant trait. And then I wanted to show you this quick, this is just from BuzzFeed, showing a few different human traits. So if you cross your hands, do you put your right thumb on top or your left thumb? I'm a right thumb.
Can you see by the dawn's early light? Ew.
Okay, So we're going to conclude there, and thank you for joining me today for this video.